It's biology with Mr. B. Biology with Mr. B. That's me! So, hello and welcome to a video going through the topic, the GCSE topic, Enzymes and Digestion. So I've got 20 minutes, and I'm going to try and get close to 20 this time, to focus on ideas that are covered in this topic, ranging from enzyme structure and linking that to enzyme function, the lock and key model. I'm going to make a big focus on factors that affect enzyme activity, because that relates really nicely to one of the required practicals from this topic. Specifically, we can look at enzymes within digestion and how they work, as well as other substances in digestion, such as hydrochloric acid and bile's function. And then there's actually a second required practical in this topic about different food tests and how you do them and how you identify them. So, first thing to focus on is, what is an enzyme? What's its job? And how does it actually go about doing its job in the way that we want it to? So firstly, I hope you don't mind my drawing. This is Eddie. Eddie is an enzyme. I'm going to give Eddie a hat. It just feels wrong not to give Eddie a hat. So Eddie's shape is very important to him. Eddie's shape is incredibly specific. In particular, the shape of what I've drawn as like a mouthpiece, but it's actually known as an active site. And the active site is the most important part of our enzyme, because it is in the active site where every single chemical reaction in living things occurs. Inside that active site, Eddie the enzyme gets to be a biological catalyst, which means it can speed up chemical reactions. Because again, linked to some chemistry knowledge, that's what a catalyst is. A catalyst is a molecule that speeds up chemical reactions, but enzymes are biological catalysts because they do it in living things. So an enzyme is a biological catalyst, which means it speeds up chemical reactions in living things. And it does so in a very, very specific manner. Now, I could link this really nicely to an inheritance topic and the idea of protein building, but I'm going to save that. But I'm just going to remind you that enzymes are made of proteins. And the fact they're made of proteins is what gives them such a specific, unique 3D shape. And the other really useful fact to know about enzymes: you'll never find them in the chemical equation, because they never get used up in a chemical reaction. As my biology teacher once said to me, they can go all night long. Yep. Yes, he did. Anyway, I've said it to you now, so passing the torch. So these are our key ideas, our key facts about enzymes. Biological catalysts, so they speed up chemical reactions in living things. Their shape is incredibly specific, in particular the shape of the active site, which is where these reactions take place. The reason why it's specific, and you can link this to the idea of inheritance and mutations, is because they're made of proteins, and they also never get used up in a chemical reaction. So, what I'd like to show you, and I'm going to try and use my amazing drawing ability, is how they actually catalyse chemical reactions. So I'm going to try and draw an example of digestion, where starch is broken down into glucose by the enzyme amylase. So I'll show you my enzyme. This is an amylase enzyme. I'll give it a little A for amylase. And a little hat. This one can have a bit more of a circle hat. Oh, it looks like a chef hat. Who knows? So there's my amylase enzyme. And I'll draw starch as this red large molecule here. Because remember, starch is a large, insoluble molecule, ready to be digested inside your body. So the first thing that has to happen is the starch, which we call the substrate, will actually bind, in this case, to the amylase enzyme's active site. So the very first step here is that the substrate, which in this example is starch, binds to the enzymes, which again, in this example I'm drawing, is amylase, active site. The reaction, when they are now bound together, will take place. And this reaction, guys, is a digestion reaction. So what's going to happen is the enzyme is going to remain completely unchanged, even though I am clearly drawing him slightly differently each time. He seems to have shrunk. Never mind. But I promise you the enzyme would be completely unchanged. But the starch molecule will be broken down. So in this case I've depicted it being broken down in two, turning from a large insoluble molecule into small soluble glucose molecules. And we call these new molecules here products. So the substrate is what bound to the active site of the enzyme. And the products are what is formed, and the products get released. Now, this is an example of digestion where I'm breaking something down. Please remember, enzymes are involved in all reactions inside your body. They speed up all of them. So they also speed up synthesis reactions as well. And a synthesis reaction, imagine the arrows the other way around. Synthesis reaction where you take small molecules and build them up. It's a reaction that requires energy from respiration. Okay, so this method, which I'm just going to leave with a diagram now, is known as the lock and key model. The reason why is that the only way enzymes function is if the substrate is a complementary shape to fit the specific active site. So if I redraw my enzyme and it's got this sort of, that sort of circly sort of approach to the active site there, a substrate that's a square shape will not fit. It would need to have a substrate that is a perfect fit into that active site. So we would say that the enzyme's active site has a specific shape complementary to its substrate. So if there are 30,000 different substrates in the body, you'll have 30,000 differently shaped enzymes. Okay. So the next thing to focus on is what factors affect the rate of enzyme activity. So there is a classic graph which they like to draw to try and force you guys as students to both describe and explain what is happening. So I'm going to focus on temperature as my graph. So I'm going to have temperature on the x-axis. I'll start at 0 degrees here and I'll go up to 100. And on the y-axis, I'll just put rate of, tell you what, I'm going to do rate of respiration. Because after all, respiration is a chemical reaction inside living things, and therefore it must be controlled by enzymes. So when I say rate of respiration, I may as well be saying rate of enzyme activity inside the mitochondria. If it was rate of photosynthesis, that again is a chemical reaction inside living things, so that rate of photosynthesis means rate of enzyme activity inside chloroplasts. And the shape of the graph we would always get would be this. And that shape would be true. It wouldn't matter what living thing you're looking at. The shape would be true. The actual numerical values might be different, but we'll always get this up and down shape if we're looking at rate. So, describe and explain. Do check out my video on the differences between the two, because very different if you get asked to describe this graph, explain the graph. If you describe the graph, say what you see and give data. Between 0 and 40 degrees, the rate of respiration increased by whatever to whatever numbers it did. The optimum is here, and then it decreased from this degree to this degree. If you're asked to describe, you don't need to use science. If you're asked to explain, then you have to use science, and it's got to be correct. So there are two parts to explain here. You need to explain why at low temperatures there is little to no reaction, why enzymes aren't working at lower than the optimum, why enzymes begin to work faster as the temperature increases, up to the optimum temperature, which is that point there, optimum. And then why enzymes, again, start to slow down their activity and completely stop their activity at extremely high temperatures. So let's start down here. At cold temperatures like this, the enzymes and the substrate will have little kinetic energy. That's the movement-based energy. So there'll be very few collisions between the two, so you'll get very few reactions. The substrate has to fit into the enzyme's active site, and they have to do that using their natural kinetic energy. If they don't have kinetic energy, they will move really slowly, and therefore you'll get far fewer reactions. So as you increase the temperature, you get more kinetic energy. I'm just going to put Ke to demonstrate that. And therefore you're going to get more collisions between substrates and active sites, so you're going to get more reactions. So that's what we explain the left-hand side of the graph. The right-hand side, we explain using a key term and the definition of that key term. At extremely high temperatures, the bonds between the proteins, between the amino acids of the enzyme begin to break. And that specific shape of the enzyme will begin to unravel. And the active site will no longer be there, and it will no longer be specific to the substrate it binds to. We call this effect of high temperature denaturing. And if you write in an exam, oh, there's less reactions at high temperatures because the enzymes are denatured, well, it's a very good start. But because denatured is a key word, you then have to explain and what that means. So what the examiner is looking for, if you ever use the word denatured, they're looking for the idea that the active site changes shape so the substrate cannot bind. Because enzymes are reliant on that specific shape active site. If it changes, because the high temperature breaks the bonds between the amino acids that make up the protein, the substrate will no longer fit to it and you won't get any reactions. So this graph is beautiful for explaining enzyme activity and what temperature is doing to it. I'm just going to change the graph ever so slightly. I'm no longer going to use temperature on the base. Instead, I'm going to focus on pH. That'll do. So I'll go from pH 1 up to pH 14. And I'm going to draw actually a couple of different graphs in different colours. So I'll draw that same sort of shape graph as before, where the optimum is around pH 7. I'll draw a graph that's going to be very, very similar in red, but its optimum is going to be about pH 8. And then I'll draw a graph in black that's going to look very different. So, with the exception of the optimum pH for these enzymes, either side of them, the reaction rate slows. And that is because enzymes denature if placed outside of their optimum pH. So, unlike temperature, which was only high temperatures denatured, pH, if it's not the optimum, it just denatures. Doesn't matter if it's too acidic or too alkali, it will just denature. And this graph here should give you clues as to what enzymes you're looking at, or at least what body part you're looking at. So if these were all digestive enzymes, I'd be able to quite happily tell you the one which is a pH of 7, well, the only organ in digestion that has that optimum pH is the mouth. So that is the enzyme salivary amylase. pH 8, that's where like the optimum pH of your small intestine and the sort of where all those enzymes go in. Now, I won't be able to work out exactly which one, but I certainly know it's the small intestine, so it could be pancreatic lipase, it could be pancreatic amylase. If you remember, the pancreas produces all the different digestive enzymes and pumps them into the small intestine. So it could be certain types of protease enzymes as well. pH 2 then, there's only one body part that is optimum is that acidic, and it's the stomach, because the stomach contains hydrochloric acid. Not to help in digestion, by the way. The acid's to actually kill any pathogens that might end up in your food. But it has a pH of 2, and therefore if you want an enzyme to work in it, the enzyme has to have an optimum of pH 2, and that's a protease enzyme in the stomach. So that is certainly one thing I'm unfortunately not going to be able to go through today, because I'm coming up to my 20 minutes already. I'm not going to be able to talk about the exact specifics of what enzymes are found in which digestive organs. So please use my revision booklet to get that information. Okay. So, I think I'll do one more thing, because it comes up fairly often and people don't like it. In digestion, there are a couple of molecules that help but don't, well, which aren't actually enzymes. Bile and hydrochloric acid, neither of which are enzymes. But both of them are essential for enzymes to work. So hydrochloric acid in the stomach, obviously it provides an acidic environment to kill pathogens. Its main role is to stop communicable disease from getting into your body. Bile, which is made in the liver and stored in the gallbladder and released into the small intestine, into the duodenum, which is the first part of the small intestine, the part just underneath the stomach. Bile is actually slightly alkaline. So it actually neutralises acidic food from the stomach. Because think, in the small intestine, in the duodenum, all those enzymes have an optimum of pH 8. So if acidic food comes down from the stomach, it would denature those enzymes. So bile, which is pumped also into the duodenum, just underneath the stomach, will neutralise that acidic food and make it pH 8 so the enzymes can work as effectively as possible. The second thing bile does as well, bile emulsifies fats. It does not break them down though. Only enzymes break down food molecules. In this case, it would be the lipase enzyme. But bile does help. When we say emulsify fats, what we mean is fats like to stick together as big droplets. Bile emulsifies it, which means it unsticks into lots of little fat droplets. Having lots of little droplets increases the surface area of the lipid, of the fat, which means more enzymes can act on it per second, which means you get more fat breakdown. So do look out for that, and do look out for the fact that when you do break down fats, you make fatty acids. If ever you're looking at a bit of data from like an experiment and digestion of fats has been taking place, and it seems to be getting more and more acidic, it's because fatty acids will be being produced. Right, so thank you very much for your time. I've gone 23 minutes, so thank you for watching. Please use my revision guides for other ideas in this topic that I've not been able to go through, but I hope that was useful. Take care, see you soon. Bye.